Assalamu alaikum. So today I'm going to continue with where we left in your last class, where we were discussing about the next generation probiotic and the gut microbiome. Here I'm starting all over again, just to bring everybody on the same page. And what we know up till now is uh, we know about the probiotics, that how they are beneficial for the health of the humans as well as other animals. And probiotics, along with their metabolites, also confer health benefits. And in this figure over here, you can see the health benefits which are conferred by the probiotics along with the postbiotic, which are the metabolites produced by the probiotics, including the short chain fatty acid, bacteriocins, and other antimicrobial products. The prebiotics, which are the non-digestible carbohydrates that will facilitate the growth of the good bacteria and the gut microbiome inside your body when taken. So they will facilitate the growth and proliferation of these good bacteria. And along with that, we have FMTs, which we call as fecal microbiota transplantation. And what we mean by fecal microbiota transplantation is that we are going to transplant the gut microorganisms from one individual to another individual. And they are available in the form of capsules, as you can see in this figure. So all these, so FMTs is still on the third trial globally. If we just see the studies are being done, and number of organisms are being tested, so that they could be transplanted, and they could be used beneficially and therapeutically for the for benefiting the gut of the humans, right? So here in this figure, if you see, along with the these four uh, beneficial attributes here, FMTs, prebiotic, probiotic, and postbiotics. We also have another terminology mentioned over here is nutribiotic. Now, the name says nutribiotic, which is like nutraceuticals, means food supplements, which are also having probiotic inside them. So we have a supplement containing food supplement along with probiotic. We call them nutribiotic. Even we call them nutraceuticals. And then we have another terminology over here, pharmabiotic. And by pharmabiotic, we means those chemicals and those drugs which are basically used for therapy, for curing any infection or any condition. And they also contain probiotics. So if they contain probiotic, so along with that chemical or drug, we call them pharmabiotics. Then we have along with them another terminology. And in this way, there are many other terminologies which are coming into being. So we have psychobiotic. Again, the terminology and the name and the word tells you that psycho comes from psychology, means something related to your brain, and biotic comes from the probiotic. So psychobiotic, by psychobiotic, we means that they are those probiotics and those products of the probiotic that can alleviate a number of conditions which are, or altered conditions of the nervous system or your brain. So they can benefit your psychological condition like anxiety, depression, or ADHDs, so schizophrenia. So there are many other disorders which are being explored, that whether the probiotic are able to uh, alleviate these conditions or not, and they're turning out to be good, right? Then, so these things we've already discussed in class, but still, since I'm starting all over again, and I'll be sending you this lecture to the Google Classroom, so that you can just take help from this if you are not understanding on your own. So in this figure again, on below here, you can see a number of disorders and number of conditions by which the infants are being affected, or the like a number of infants are colic, or they have a number of infections. And we have children who are suffering from diarrhea or antibiotic associated diarrhea, and lactose intolerance, and then we have inflammatory bowel syndrome, and so many dermatitis, all of these conditions. Then we have a number of conditions in adults, which also again include lactose intolerance and antibiotic associated diarrhea and UTI or traveler's diarrhea, vaginal infections, and so many others. So all of these conditions, whether they are infants, children, or adults, they can be treated and they can be managed by using probiotics, right? So we can use probiotics either along with food supplements. We can use probiotics either along with any pharmaceutical like pharmabiotic. So they could be giving you a good benefit. So basically, what the probiotic do when we give them, they um, they can change the conditions in your inside your body. Like if in certain conditions our body is suffering from dysbiosis, means that the pathogenic organisms are more in number, and naturally if the balance goes out, they would be causing that infection, they would be showing the pathogenesis. So when you give a number of good bacteria and the increase the number of probiotic inside your body, what will happen? It will reduce dysbiosis. It will again maintain a balanced gut and a balanced microbiome, which will give you an improved health and prevent you from the disease, right? So this is what we've already discussed. Again, metagenomic. So things have become easy and things have become rapid. We've discussed a lot about metagenomics, but again, I'm telling you that I'm repeating these things so that you have a thorough understanding. So, metagenomic approach is the approach which is happening currently, which is used in this time, and such a, I mean, this approach will give you maximum output, numerous data, a lot of information and knowledge. How? Just using the stool sample. What is done? We use a stool sample from any person, whether it's a patient or a normal individual. We isolate the DNA from a stool sample, and then simply we do the next generation sequencing, NGS. What does the NGS do? It gives you the sequences of all the types of organism which are present in the stool, right? So all the DNAs are being analyzed. So, for example, 20 million sequences being analyze, and it gives you all the sequences, right? Then a number of sequences, the known sequences, but what do we mean by known genome? A number of sequences are already present, uploaded in the databases. So the sequences which you have got from the stool sample, which are still unknown, and the one which are already present in the databases would be compared, would be aligned, would be seen that which one is matching to which known genome, and this will give you a clear idea about the type of organism, or it will give you the identification of the organism that you're looking in the stool sample. So it will give you the whole uh, i mean data, giving you a complete sequence identification of the organism present in the stool sample, right? So it give you a prediction map, it will give you the lot of tools in bioinformatics, statistical analysis and identification. So finally you come to know that there are these many organisms which are present in the stool sample, right? And if you see it therapeutically, if you take a stool sample from the normal individual and you take a stool sample from a disease or a patient, right? So you'll see the difference in the type of species and genus which are present in the stool sample. So the number of the beneficial organism would be reduced in case of the patients or those who are suffering from any disease. So, what will we do? We'll give them such prebiotic and probiotic, which will facilitate the growth of the number which have been reduced. And finally, when the number increases, so the body will return back to a balanced gut microbiome and it will return back to homeostasis. So, these are the rapid and the very accurate uh, analytic tools. So, we call them metagenomic approaches, right? So see, you're getting so much of information from metagenomic. What are we getting? Let me just zoom in. Now in this figure, what you see, right? Uh, this is what is showing you the gut from inside, and then this is showing you your gut microbiome, the organism which are present in your gut. So we've analyzed them by metagenomics, okay? That I just mentioned you earlier. Now from this analysis, a lot of information has been generated. What information you're getting? You're getting the information about the genes which are present, by the DNA which is present in a stool sample. When you know when you have the information about the genes, you know that which metabolic pathways are being encoded or being um, you can say managed by these genes. Then we also know that how many genus or species have been reduced in number and creating dysbiosis. So we know this functional dysbiosis. We also find out a whole lot of communities which are present on your gut microbiome. So that is diversity. So you find out that a lot of diverse population of different genus of microorganisms are present in the gut, right? Then we have interaction between host and microbiota. So as we studied in immunomodulation, if you remember, I always showed you a figure which is showing you the intestinal lining, the mucous membrane, and how the microorganism, when they come to the mucous membrane and they interact by means of the toll-like receptors and produce signals inside the cell or underlying the mucous membrane. So this is the host and microbiota interaction. It could be the interaction between the pathogen and the host. It could be the interaction between the probiotic and the host, right? Then we can also know about the co-evolution of host and microbiota, that how they survive and how they respond to each other. And finally, we also find about we have a knowledge about antibiotic resistant genes. So when we do the metagenomic and we know about all the genes which are present in the stool sample, any sample which we are utilizing, so we will be knowing the genes which are uh, I mean, uh, encoding for the antibiotic resistance. And we know that any probiotic should be susceptible to antibiotic in order to pass the criteria for being a good potential probiotic. We should not have antibiotic resistant genes in a strain which is being used as a probiotic, right? Why? Because it will be horizontally transferring the gene to the pathogens, and then the therapy would be difficult and will require antibiotic resistance. Now, in this figure, after doing the metagenomics, you get these type of graphical data. And what do you see over here? You get a phylogenetic tree over here. And you can see, again, I'll just zoom in. Okay. Now, in this phylogenetic tree, what you're seeing that how many different types of genus are present in the gut microbiome. We have Bacteroides, we have Proteobacteria, Acinetobacter formicatus, then we have other bacteria, Streptococcus, we have Helicobacter, we have Lactococcus, Lactobacillus, and so many others, right? Akkermansia. So these are the different genus which are present. There are many more. So in a population, we have about 1100 species and 10 million genes, right? So this is the statistical figure shown over here, that in each individual, at least there are 160 species and there are 540,000 genes. So this means that we almost share 40% of the genes amongst individuals, right? So all humans share 40% of genes amongst each other, and they vary in 60%, right? Now, this figure here shows you, wait, this figure shows you the um, benefits of the prebiotic and how you can tailor a prebiotic, how you can prepare a prebiotic. And if you just recall your memory, what are prebiotics? Prebiotics are the non-digestible carbohydrates that will be facilitating the growth of the gut microorganism or the beneficial microorganism inside your body, even the probiotic. We want an increased number. So we give such a diet, such a raw material inside your body, which is non-digestible, means it will not be degraded in the stomach, neither it will be degraded in the small intestine. Straight away, it will reach in your large intestine, where it will facilitate the growth of the beneficial bacteria, right? So this figure is showing you all those raw materials from where we can prepare prebiotics, which include bacterial exopolysaccharides, means a number of bacteria produce exopolysaccharides. Then we have algal polysaccharides, and then we have plant polysaccharides. So polysaccharides are large macromolecules. In order to prepare a prebiotic, we need to hydrolyze them, we need to degrade them. So we use a number of enzymes to degrade them. So hydrolysis of these polysaccharides in order to get monosaccharides or oligosaccharides. So these oligosaccharides, which are coming by the hydrolysis of oligosaccharides, are being utilized for the to have prebiotic property, so and for the facilitation of the growth of the gut microbiome. So these prebiotic properties include anti-adhesive property, novel structure diversity, species specificity, and enhance persistence. Kiwa. Okay. So now, just comparing the probiotic from the prebiotics, we already know what are probiotics, beneficial bacteria, which when given in appropriate number, adequate number, which is around like 10 to the power 6 and above, it will confer a health benefit. And the prebiotic basically are the oligosaccharides, and they are the non-digestible carbohydrates, tacos, and glycanases and galactosidases. And we studied a lot of prebiotic as well. Some are synthetic and some are natural. This is a timeline showing you the discoveries, and finally, ultimately, we come to the next generation probiotics. Starting from 1957, Louis Pasteur found the spoilage organism, and he thought at that time that lactic acid bacteria are spoiling the food, but on the other hand, it was found that they were able to ferment a number of food products. So he found out the fermentation property of the lab. Then throughout that time period, after about like a couple of decades, in 1878, the lactic bacteria were isolated from milk by Lister. And then again, around a couple of decades further, Metnikov described Bacillus bulgaricans, which is associated with health benefit. So, with that, started a new era, and people from time to time isolated a lot of lactic acid bacteria from a number of products, food products, natural products, dairy products. It included Lactobacillus casei, Bacillus acidophilus, Bifidobacterium, and so many others. If you just come to further to 1989, this was a time when Fuller defined probiotic, and he gave the definition of probiotic. It's not that people did not know, did not discover the organism which are now being called as probiotic. They were already there, like Bifidobacterium and Lactobacillus, as you can see in 1889 and before that as well. But the term probiotic and the understanding about the probiotic was first introduced and given by Fuller in 1989. And after that, in 2001, WHO recognized the definition of probiotic. FAO and WHO recognized as probiotic. So these two timelines are important, and we have discussed about these two timelines in 1989 about the Fuller, and 2001 when the definition of probiotic was given by the WHO. Further on in 2003, everybody knows that the molecular era started in this century, in the beginning of this century, and human genome was also sequenced. Complete human genome was sequenced by 2003. So, as the human genome was sequenced, the sequences of other bacteria were also being analyzed. So, the genomic era first analyzed the sequence of a probiotic Lactobacillus plantarum. We all know that Lactobacillus plantarum is a very potential probiotic which we have studied in terms of alleviating the severity of COVID or SARS-CoV-2, right? And in this timeline, last of all, 2016, FDA gave the guidelines for live biotherapies. So we'll see what is the difference between live biotherapies. They're also including the good microorganism or the beneficial microorganism. So I think there's no need to again define or explain you the definition of probiotic, which you already know. You can have a look. Everybody knows about it. We know what are prebiotic. So again, I'll skip that, right? Non-digestible carbohydrates, especially the oligosaccharides, fructooligosaccharides, galactooligosaccharides, lactulose, and inulin. And we've studied in detail the prebiotics and what are the benefits of the prebiotics. Next generation probiotics. So this is all what we've started. What are next generation probiotics? So WHO and FAO has defined them as the non-traditional microorganism. How they're different from the traditional probiotics? See, uh, of all these years, people are being using these traditional probiotic, being isolated from the dairy products, being isolated from other natural sources. But naturally, a time comes when the utility of these traditional microorganisms becomes saturated. So we need to explore more resources, more options and avenues from where we can find more organism. Like we have antibiotics and then next generation antibiotics. So we have probiotics and next generation probiotics. So these are basically the non-traditional microorganism. Mostly, these belong to the organism which are present inside our body, behaving as our gut microbiome. So how we take the next generation probiotic, comparing them with live biotherapies, LBPs. So we know what are probiotics and next generation probiotics, but the live biotherapies, basically we are using these next generation probiotics for therapeutic purposes, right? It includes Akkermansia, Bacteroides, Faecalibacterium, Firmicutes. All of these genus are present inside our body. So we are utilizing the benefit of these organisms for therapeutic purpose. I hope you're understanding, right? So they contain live organism. We all know that they are used for the prevention and treatment and the cure of a number of diseases and conditions, but they are not used as vaccines, right? So we can isolate them, identify them, characterize them, and then produce them, do the clinical trials. And after doing the clinical trials, we can utilize the benefits of these organisms, right? Now, here I'm showing you some charts or some tables which have just been summarizing the studies that have been conducted recently or in the current times. And still, a lot of work is needed to be done to understand these bacteria, these probiotics, I mean, before utilizing them on humans, right? So if you see in the table, the name of the organism is written, and then the nature, or means, the type is written, means whether they are from human origin or they're genetically modified, then which disease are they targeting on, and what work is being done, the experiments, right? So one of the next generation probiotic that is being explored and studies are being done on humans. We all should know that whenever we explore and whenever we study any organism and we want to utilize it on humans, bring it into the market and utilize the benefit of it, we need to first um, study it in vitro in the lab. Then, after characterizing it, finding out that it has a potential, then we explore it in vivo on cell lines and on animal models, and then we utilize that into human volunteers, and then we come to utilizing them on the public or on population, right? So all of these studies which are being done on Bacteroides ovatus, xylem solvens, Bacteroides ovatus, different strains of Bacteroides ovatus, and Dory. So these studies have been done on mice. So they're still on the first trial. Some of the studies have been done in humans as well. Uh, Number of Bacteroides are isolated from humans, which you can see Zalani, solvents, ovatus, and the last one, Dory. They are isolated from humans, where the other two are genetically modified organisms. So basically, the technology of genetically modification is utilized for benefits, but you never know that whenever you are editing the genes, whenever you are altering the genes of any organism, it might turn out to be much pathogen or much detrimental, uh, other than being giving you a positive result. So there are always um, risk factors associated with a genetically modified organism, but this technique has been adopted always for the benefit side, right? All of these organisms are targeting cancer and inflammation and heart diseases. So we know that the these probiotics how do they manifest cancer? Do remember, whenever any organism, if we say probiotic is inhibiting cancer, or it is good against cancer, this means that it is inducing apoptosis in cancer cells so that they undergo lysis and they are damaged, and they reduce the proliferation of cancer cells. So this is what the objective of the probiotic is against cancer. Do remember that they will um, decrease the proliferation of cancer cells and increase the apoptosis of the cancer cells, right? And the level of evidence that they've done on mice and what they have seen. So this is what they have shown you in this figure. So we are not going into the detail of every organism because still they are on a preclinical trials. So we are just having a summarized view of all these organisms. In this second list, you can see number of other next generation probiotic, including some more strains of Bacteroides fragilis, acidification. Then we have Clostridium butyricum, Faecalibacterium prostacea is a very important next generation probiotic. Then we have Lactococcus lactis and another strain of Lactococcus lactis. Most of them have been isolated from natural human source, mouse, which is also a natural source. And the last two are genetically modified and they have been isolated from food sample. What they are targeting, if you can see the target disease, many of them are targeting inflammation, and you can observe inflammation in a lot of infections, including cancer. Last two genetically modified are targeting allergic symptoms or autoimmune diseases as well. Even they are targeting type 1 diabetes. Further on in the slides, we will be also seeing the names of those probiotics which are reducing the conditions, or you can say alleviating the conditions of diabetes, right? So the experiments or the evidence which they have got from the experiments, uh, what you can see over here, they have written low evidence, low to medium evidence. This means that not much extensive evidence is there. Why? Because they are still on in vitro work, in vitro. So the preclinical trials are still on the in vitro, so they require you can, because it's low level. So in vitro. So after in vitro, they need in vivo work. They need on animals. They need on humans. So a lot of work still to be done. And low to medium evidence gathered for claims in humans. So they have done some sort of work on humans as well. Same goes for the others. Now comes a little bit of a specific details or specificity or specific function of one of the next generation probiotic, and the name is Akkermansia mucinifera, a very important probiotic, and which is called as a next generation probiotic. Why? Because it's present inside your gut microbiome. Do remember, Akkermansia mucinifera is an organism which is present inside your body, right? Behaving as your gut microbiome, and it always benefits your body. How it benefits? Now, it is seen that in one of the study, it was seen that people who are suffering from obesity, right? All those people who are suffering from obesity, and even the animals, when they did the study on animals first, so all those obese animals and humans showed one type of a genus very dominant. And that one type of genus was Enterobacter clausii, right? So this genus was, outnumbering the other good bacteria in people who are obese. And in obese people, they found that the number of Akkermansia mucinifera, the good bacteria, the number of Akkermansia mucinifera is lowered. It's is less, right? So now what does it indicate? It indicates that since there is dysbiosis, Enterobacter increase in number, mucinifera decrease in number, which is not a balanced microbiota. So what they did, they give a treatment, a diet containing oligofructose, which is a prebiotic. And due to this oligofructose, the number of Akkermansia muciniphala increase, right? When the number of Akkermansia muciniphala increase, this resulted in a improved metabolic profile. How? Means that it decreased the number of Enterobacter clausi, right? And secondly, it also showed or it also improved the sugar metabolism. So when the sugar metabolism was increased, when the we can't don't say sugar metabolism, sorry, it's glucose metabolism. So when the glucose metabolism was improved, when the other metabolic activities were improved by increasing the number of Akkermansia muciniphala, what happens that the obese mice return back to its normal condition. So obesity was reduced. Obesity was it was seen that obese mice were showing a improved profile, right? So even when we increase the number of muciniphala, it can decrease the metabolic endotoxemia and also inflammation in the adipose tissues. So they are also improving the gut barrier function, right? Next is again, if we see the benefit of these probiotics, next generation probiotic, let me just enlarge a little bit. Okay, okay. Now, what this figure is showing you, you have to concentrate on this figure because it's very important. Here you can see this is the intestinal lining. This is the mucous membrane. These are the epithelial cells, okay? We've seen this such type of figures before also. And here you have the mucous membrane, mucous layer, the yellow path which is shown over here. And on the top you have a number of organisms being shown, right? Some of these organisms may be pathogens and some are the normal flora. And we include those which we are giving to the person, to the individual for the live biotherapy. So if we are giving probiotics or next generation probiotics as live biotherapy, including lactobacillus species, bifidobacterium species, Saccharomyces boulardii, and Akkermansia muciniphala, what are the changes that are going to happen, or what are they going to induce inside the intestine, right? That is going to change the metabolism. Are you getting me? That we are going to understand that what are those signals and what are those changes being induced by these organisms which are being given orally in terms of next generation probiotic. Why are we calling them next generation probiotic? Because it includes Akkermansia muciniphala. It's including lactobacillus bifidobacterium, which are the traditional probiotics, right? So what are the changes? Now, number one change here, if you just see in the enterocytes are the epithelial cells of the mucous membrane, right? So if you just see one enlarged view of the enterocytes, see BSH, bile salt hydrolase activity. Now, this is an enzyme which can deconjugate bile salts, bile acids, right? They can deconjugate the bile acids. And by doing that, what are they doing? They are reducing the cholesterol level. Now, how are they reducing the cholesterol level? All of these organisms, whether they are the traditional probiotics, whether they are the next generation probiotic, they have the ability to produce an enzyme bile salt hydrolase. This bile salt hydrolase enzyme has the ability to reduce the toro beta MCA, and this is the type of bile acid, right? What does it do? It deconjugates it, means it removes the amino acid from the bile acid. Usually, the bile acid, when they're called as conjugated bile acids, they are quite soluble, right? So when they are conjugated bile acids, they are not good for our body. Why? Because they will be converted to cholesterol. As they are soluble, they would be absorbed into your bloodstream and increase your blood cholesterol level. So what we want to deconjugate the bile acids. When we deconjugate the bile acid, the taurine or the glycine is removed from the bile acid, and this produces a signal inside the cell that will reduce one of the signaling receptor inside the cell as FXR, farnesoid X receptor. This will reduce the levels of FXR. This reduced level of FXR will reduce the level of FGF, which is an enzyme which is required. It is an enzyme which is required to increase the level of cholesterol, right? I hope you people are understanding. I'll repeat this, but let me just finish this, how it is reducing the cholesterol level. So when there is a FXR reduction, right, it also decreases the level of TGF, which is dependent on FXR. So FXR reduces. So first, BSH is produced from these organisms. The type of bile acid is deconjugated, right? The amino acid is removed from it. Either glycine or taurine is removed from the bile acid. This gives a signal to the FXR, which is the farnesoid X receptor. That reduces FGF, right? An enzyme which is giving signal to the liver. For what purpose it is giving signal to the liver? As you can see, this line is extending to the liver, is giving signal to the liver to decrease CYP7A, right? Now, this is an enzyme whose under normal condition, when this decreases, cholesterol level increases. But here, what is happening? FXR is reducing, level of FGF is reducing. This gives a signal for an increase opposite. This happens an opposite. Okay, please understand this. It increases the CYP7A1, right? When this enzyme activity increases, cholesterol level decreases, triglycerides decreases, bile acids decreases, all the bad fats, all the not required fats level decrease, right? Normally what happens when there's no bile salt hydrolase enzyme, there is no deconjugation of the bile acids. The conjugated bile acid will increase FXR, will increase FGF, and decrease CYP. This decrease in CYP will increase cholesterol. This is what normally happens. And this will lead to an increase in cholesterol level in the blood. But actually, when the BSH bile salt hydrolase enzyme produced by the probiotics will deconjugate the bile acids, removing the glycine and the taurine from the bile acid, now the bile acid will be less soluble. See, do understand, when anything which is less soluble will not be absorbed in the blood. So after deconjugation, the bile acids become less soluble and it will be removed from the body through the feces, right? But when it's conjugated, it's more soluble. It can increase the cholesterol level. So what the BSH is doing, one last time I'm repeating, it's deconjugating the bile acid, decreasing the level of FXR, which in turn is decreasing the level of FGF. And this decrease in level of FGF is increasing the level of enzyme SYP in the liver, which will give signals to reduce the cholesterol level and other fats, triglycerides. Okay, this is one side of it. Then on the other hand, the probiotic produce, as we all know, they produce short chain fatty acids, right? Butyrate. These short chain fatty acids will produce signals to the GPR443 receptor on the enterocytes. When this receptor gets signals, the cells start to produce GLP, an hormone, a peptide. GLP is glucagon-like peptide 1, glucagon-like peptide 2, right? And what is the glucagon-like peptide 1 doing? The glucagon-like peptide 1 is increasing insulin sensitivity and it is reducing insulin resistance, right? So it's reducing insulin resistance. This means that now the insulin inside your body will work to metabolize the glucose level. Secondly, the glucagon like P2 is another hormone which will be, it is antimicrobial towards a number of pathogens, so it will produce antimicrobial activity. And also, it will maintain a good gut barrier function, right? So, uh, then we have the third one, the enterocytes or the epithelial cells of the intestine produce another antimicrobial which is called as REG3 gamma. The level of REG3 gamma increases, and what does this do? It is basically an antimicrobial peptide which will help to fight against pathogens, and also it maintains a good thickness of the mucous membrane. So this will maintain a good barrier function. So you can see that how different organisms are maintaining the health of your gut, right? So do understand this slide very carefully. Now here, more benefit of Akkermansia mucinifera, which I told you that is a very important next generation probiotic. And we've seen that how it can reduce obesity when we increase the number of Akkermansia. One side of this figure is showing you a disturbed gut where you can see pathogens, where you can see thin mucous membrane, where you can see the permeability of the mucous membrane is disturbed or damaged. Why? Because the tight junction proteins, occludin and claudin, their level are decreasing. So, and all of this is happening because of gram-negative organism or the endotoxin from the gram-negative organism, the LPS. So this is the condition. So this is called as dysbiosis, as you can see, pathogens as well as other organisms. On the other hand, when you keep on giving the therapy, the live biotherapy of next generation probiotic, in terms of Akkermansia mucinifera, this means that we are increasing the number of Akkermansia mucinifera, right? So what will happen? They will outnumber the other organism. They will interact with the toll-like receptors which are present on the mucous membrane epithelial cells. Increase the level of tight junction proteins. You can see these locks here. Gaps have been reduced, which you can see over here. So they are increasing, they are reducing permeability, or they're maintaining permeability of the intestine. They are maintaining a good gut barrier function, right? Reducing inflammation. And you can also see another thing, that the mucous membrane thickness has increased. There's also inducing signals that is propelling the epithelial cells to produce mucous membrane, right? So overall, if you see, hepatic steatosis is reduced in case of a treatment by Akkermansia, means a fatty liver is reduced, hepatic inflammation is reduced, insulin resistance is reduced, inflammation is reduced. We know how inflammation is reduced. Remember, when we studied immunomodulation, that the probiotic produce short chain fatty acid that will produce signals to induce those cells that produce anti-inflammatory cytokines like interleukin 10 that will reduce inflammation. Even they reduce type 2 diabetes. And in the previous figure, we've seen how they reduce diabetes, that they produce GLP-1, which will reduce the insulin resistance and maintain a good glucose metabolism, and that will give you a good gut uh, health, right? Improve the metabolic disorders as well. Next, next generation probiotic is Faecalibacterium prasnozae, another important one. And the role of prasnozae was studied in vivo as well as in vitro. And specifically, it is involved in reducing inflammation. So it's able to reduce the number of activity of the T helper 1 and T helper 17 cell that we know that produce pro-inflammatory cytokines. And how do they do that? By producing interleukin 10 secreting cells. So this means that the Faecalibacterium prasnozae is basically focused on immunomodulation or enhancing the immune responses and reducing inflammation, right? The third next generation probiotic which we need to study is Bacteroides fragilis and uniformis. Now, what does Bacteroides fragilis do? So basically, it produces an exopolysaccharide, PSA, and this PSA is having immunomodulatory potential. How? Means this polysaccharide will be interacting with the epithelial cells having toll-like receptors, activate the T cell underlying the epithelial cells of the mucous membrane, and they will maintain a good immune system in the host, hemostasis in the host. So I'm not going into the immunomodulation all over again, but I hope that you people are studying immunomodulation as you study in detail in the class. Then, if we give orally Bacteroides uniformis in a mice which is challenged with high fat diet, means if we give any animal a high fat diet, and then we want to see that by giving Bacteroides uniformis, whether the fat is being reduced or being metabolized properly, whether there's a reduction in the reduction in the LDLs, the low density lipopolysaccharides or not. So it basically improves the lipid profile, reduces glucose, insulin, and also increasing the tumor necrotic factor alpha production by the dendritic cells and basically stimulating phagocytosis, right? Last of all, as I told you initially, that there's a term called psychobiotic, right? And by that, what we mean those probiotic which are used to uh, treat the neurological disorders, the psychological disorders, including autism or ADHD, which is attention deficit hyperactivity disorder, schizophrenia, anxiety, depression, so many mood swings and all. So now a whole lot of research is being done on those microorganisms, especially the next generation probiotic, that how they are going to uh, significantly alter the conditions uh, when there is any neurological disorder. So in this figure, again, I'm going to zoom in. So just focus on this figure. On the top, they're showing you the stages of life, starting from the prenatal, means at the time of birth, infancy, after that time, a few weeks after, like 40 weeks after the birth. Then we have childhood, 2 years after birth, and then adolescence, 12 to 18 years after your birth. And then we have old age, which can exceed after your 30s and 40s. It could be, it depends, right? So in all those stages, if you see in the square figure, in the cube, they're showing you the organism or the microbial community which is present in these organisms. So if you see in the prenatal condition, the individual or the child which is born does not have a very diverse microbiome. See over here, right? It's scarce. But if you see in as the child reaches infancy and childhood and adolescence, so if you just see the adolescent diversity is quite much more than the other stages. Even if you see in the old age, we are seeing a deterioration in the microbial diversity. This means that a good microbial diversity is required to have a stable nervous system, to have a stable neurological condition, right? And it is seen that in the early stages and in the last stage, you can see these are the graphical statistics which they have shown you. In the early conditions, when the gut microbiome diversity is less, the chances of neurological disorders are more, right? If you reach to the old age again, the gut diversity decreases and declines. So again, the chances of neurological disorders increase. And if you just see the development of the neurons here, Starting from the here, you see the synaptic density, which is less, and it increases as you grow in age, right? The density is increasing, right? Myelination and then demyelination starts. So we're not studying the nervous system, but at least you can visualize over here that initially the synaptic density is less. It increases in the childhood adolescence, which is the time where you have a diverse population of gut, and it reduces further on in the old age. So this figure shows you that the microbial diversity or your neural development and your neurological health, your nervous system health, completely depends upon your gut microbiota diversity, right? So, a temporal profile of the neurological developmental sequences is shown here, as you can see, and I've explained you, right? So here I stop and discussing all the benefits of some of the prominent probiotics. You should focus more on the probiotics which we have discussed here: Bacteroides fragilis, uniformis, Faecalibacterium processi, Akkermansia muciniphila, and uh, these ones, different strains of Bacteroides and different strains of you can see genetically modified organism, right? So I'll stop here. I've given you this PDF, although I do not usually give such PDFs which have slide sharing, because then what happens, you limit your knowledge to only these slides. So I want you to study more about it. I've given you another PDF also. I've given you these slides also. I'm now just posting this lecture on the Google Classroom, so you should have a better understanding of the next generation probiotic, right? So please take help of this. Even still you have any questions regarding this, you can put up questions on a Google Classroom until you have your exam on the 16th, right? So thank you so much. I'll just leave now. And since this is a recorded class, so naturally we can't have a live question answer session. I hope now you people have understood or have a sound knowledge of next generation probiotic. Thank you so much, and Allah Hafiz.